Okay, so I think that was the slide where we have finished, right? So we have seen this medullary ray, as you remember, and now we're moving on. So the next slide that we have, this is going to be kidney, Indian ink together with hematoxylin. So sorry, it's just like automatically when I type H and E. It's just forever imprinted into my brain. So it's not just, it's not with eosin, okay? So it's just Indian ink together with hematoxylin. For sure, you have seen Indian ink before. So where did we see it?
The lungs.
Very good. In the lungs. Okay, so we saw there for the pulmonary circulation, right? So we injected this ink into the um, blood vessels to see where it will take us. So the same idea is here. So the purpose of the slide is just to see the blood vessels that we talked about, and not only. So the idea is the same. So here we have the cortex, and this is going to be the medulla right here. Inside of the cortex, you can see some kind of dots. What could these dots be?
Renal corpuscle.
Of course. Okay, so these are going to be renal corpuscles. Why are they seen with the Indian ink?
Because it's
filled with blood.
The glomerulus.
Because of the glomerulus, yeah, because it does contain blood. So renal corpuscles has glomerulus. Glomerulus is just, as you remember, a knot of penetrated capillaries, right? So we do see it, and yeah, okay. So because blood vessels are there. Okay, so we magnify a bit, and as we have seen, here are the renal corpuscles, and within it we can see capillaries, which is the glomerulus. Outside of it, you can see that there's some kind of proximal and distal convoluted tubules, right? So proximal are a bit redder, distal are fainter, and between them, there's some kind of black lines. So what could they be?
Peritubular capillaries.
Very good. So these are going to be the peritubular capillaries. So this is the area where your tubules are going to be reabsorbing into, right? So we have said we will be removing and modifying your urine. Some things will be removed from the urine and they will be put into a blood vessel. Surrounding proximal and distal convoluted tubules, we have specifically the peritubular capillaries. We are here in the medulla, and you can see some straight blood vessels. So what could they be?
vasa recta.
So they are going to be vasa recta. Yeah, so they're surrounding the loops of Henle. So these are the vasa recta. You can see that vasa recta are in the medulla, but could they be in the cortex?
Yeah.
Where exactly?
In the superficial cortical.
So where do we have it? How do you call the area where loop of Henle is in the cortex?
Medullary ray.
Medullary ray, very good. So if I go back here, for example, this is medullary ray. Do you see that it has straight tubules of some sort? So these are vasa recta. If I go even more back, you can see that this is the cortex, and these are probably medullary rays, right? So we can see them here. So they are within the cortex. They do contain thick components of the tubules of the loop of Henle, right? So proximal and distal straight tubules are here. They are still part of the loop of Henle, so of course, vasa recta will be surrounding them as well. So vasa recta are not only in the medulla. You could also see it in the cortex within the medullary ray. Ureters are muscular tubes which are composed of two muscle layers. We have the inner longitudinal and outer circular layer. Anything looks weird?
Isn't it usually inner circular?
It's usually the opposite. So for example, in the GI, we saw that it was inner circular, outer longitudinal layers. But now that's the big difference for you, that in the urinary system, the arrangement of the muscles is the opposite way, right? So if you compare muscle layers in the GI and in the urinary system, you will see that it's the opposite. right? So in the ureters, it's inner longitudinal and outer circular layers. In the GI, it's inner circular and outer longitudinal layers. The epithelium there will be the urothelium or transitional epithelium, which we have seen last semester. You remember? How do you call these cells?
Umbrella.
Umbrella.
Umbrella cells. Why are they called umbrella cells?
They cover
They cover more than
Amazing. They cover two cells. So you will always see two cells under them. These cells are called piriform cells, right? So umbrella cell always covers two piriform cells. Why is it called transitional epithelium? Because the shape of these umbrella cells will be always different depending what's the level of the stretching, right? So is my bladder full? Is my ureter extended, right? And etc. Sai, can I ask you to mute yourself? It's giving me a bit of echo. Sorry. Just, yeah. Okay, so here we have the umbrella cell and here we have the piriform cell, so understandable. At the bottom we have the basal cells, which are stem cells, which will be given rise to all of the cells above. Yeah, we have talked about this part so that it can be very unique, this epithelium, because of its ability to stretch, right? So depending on how full your bladder or ureter is. Okay, wall of the ureter is a bit thinner than the wall of the bladder. So you will see that in the wall of the bladder, there will be 3 muscle layers. It won't be 2, it will be 3 of them. So that's why it's a bit thinner. Like most tubular structures, your ureter has mucosa, muscularis externa, and adventitia, the same thing. But very special that there is no muscularis mucosa, nor is there submucosal layer. So both of these areas will be absent, okay? So both of these layers will be absent. So you have already seen this picture last semester, okay? So last semester we already talked about it. So this is your transitional epithelium right here. And this is the muscle layer that can be seen, okay? So this is the muscle layer that can be seen. Outside of it is the adventitia. If you're a super professional histologist, which you are, you're going to even identify this epithelium with this magnification. You don't even need to magnify. You can even with this magnification, you can see. If you pay attention, if you're a very detail-oriented person, you'll actually see that the top layer kind of stands out, okay? It's almost like it has its own basement membrane. It looks like that, right? So it looks like it's very, very standing out. So if you magnify, of course, you will notice that these cells are your umbrella cells, that they do stand out. They're screaming into your face that look at me, I'm covering the 2 piriform cells under it, right? So these are the umbrella cells. If they are present, then this entire epithelium is going to be called urothelium, or another name for a transitional epithelium. So again, transitional epithelium possesses top umbrella cells. Underneath them are piriform cells, and at the bottom we have the basal cells, which are stem cells. It's called transitional because it can change shape. Take a look here, umbrella cells are cuboidal. Literally a bit upwards, it's already squamous. Do you see? It already has squamous appearance. So it depends, okay? All of the umbrella cells, it depends what is the level of, I don't know, what is the extent of the hydrostatic pressure that the urine is exerting. So probably in this area, there was a bit of pressure present. So as a result, these umbrella cells kind of stretched out. This area here is the lamina propria. Notice that it's not so filled with the lymphocytes. So we saw typically lots of lymphocytes in the GI within the lamina propria, but here not so many. So some fibroblasts are present, and yeah. Muscle layers are here, okay? Adventitia is outside. So nothing interesting. Ureter, urinary bladder is a distensible sac, which is temporarily storing the urine, consists of the same things. So you have mucosa, muscularis externa, and adventitia. Mucosa is the same, exactly the same epithelium. We have said that muscularis externa is a bit thicker because it actually possesses additional layer, right? So inner longitudinal, middle circular, and outer longitudinal layers. Together, these 3 layers together, or basically your entire muscularis externa has a name. It's actually called the detrusor muscle. So the detrusor muscle is your muscularis externa, which consists of three layers: the inner longitudinal, middle circular, and outer longitudinal layers. It has some kind of embryological importance. So when you do embryo, you will realize that mucosa of the bladder actually comes from endoderm. Okay, so it has endodermal origin, the same as your GI. But muscularis area of the bladder actually has mesodermal origin. So your bladder does not come from one thing. It's like a little bit of everything. So there's a bit of an endoderm there. There's a bit of a mesoderm there. So yeah. Okay, superior surface of the bladder possesses serosa, because superior surface of the bladder is not connected to anything, right? So it's free. It's just not attached to anything. So there is no adventitia. It's just serosa. As you remember, serosa consists of mesothelial cells, right? So simple squamous cells just kind of covering the organ. Inferior surface of the bladder has adventitia, because in both genders, it is connected to the anterior abdominal wall, just a bit. And you will see that in males, it's also connected to the rectum, and in females, it is connected to the uterus. So the bottom part of the bladder will be connected to lots of things. So that's why it has adventitia there. So that's the wall of your bladder, right? So we can see here that this is mucosa. Starting from here is the muscularis externa, right? Which has three layers. Inner longitudinal is this one, middle circular, and outer longitudinal. Outside of it here, we can see a bit of adventitia. Do you guys remember how is the structure called? Because we already seen it a couple of times. No? Okay, so this is called ganglion. You remember what is ganglion? What is?
It's a bunch of
Red
neurons.
fiber.
It's a bunch of cell bodies of neurons. So these blue dots that you can see here, these are cell bodies of neurons. How do you call ganglion in the central nervous system?
Nucleus.
Nucleus, very good. Okay, so ganglion is a collection of cell bodies of neurons in the peripheral nervous system. In the central nervous system, it will be called nucleus. Okay, epithelium, the same. So urinary epithelium, urothelium, transitional epithelium, umbrella cells, piriform cells, basal cells. This is your lamina propria here, so nothing interesting. Which kind of blood vessel is this?
Arterial.
It's an arterial. Why would you think this is arterial?
You have only one layer of
Smooth muscle. Yes. Okay, so it has only one layer of the smooth muscle cell. So this is arterial. What about this one?
capillary.
Capillary. What about this one?
Small.
Don't forget we have two types of venules. Well, which one is this?
Postcapillary.
Postcapillary venule. The two types of venules that we have, this is a postcapillary and muscular venule. Muscular venule will look a bit bigger. It will be double the size, and it will have lots and lots of muscles. Okay, here under the lamina propria, we have some kind of area which possesses like small veins and small artery, right? So it has this kind of area. Is it submucosa? It's debatable. In the book, it says it's not submucosa. In the lectures, it says it's not submucosa. And then you go to practicals and they will say it's a submucosa-like area. Okay, so it's a dense connective tissue with blood vessels, which is kind of connecting your lamina propria to the muscularis externa. Okay, so you wouldn't call it submucosa. So you can't technically say that this is a submucosa layer, but it's a submucosa-like layer, okay? So because it's kind of connecting the two areas. As you remember, ureter didn't have this. There was no structure like that. the layers of the detrusor muscle are here, right? So in internal, inner longitudinal, middle circular, and outer longitudinal. So together they make up detrusor muscle. This is another slide, another bladder. So here is an overview of its wall. It was stretched on purpose to show you that how urothelium changes. Do you see it's completely different right now? It doesn't look the same whatsoever. But you can still see umbrella cells. So they are looking different. They're squamous, right? But still they're covering a couple of cells, the piriform cells, so they are umbrella cells. It means that it is urothelium. It is super, super stretched, right? Lamina propria. So yeah. Okay, so we finished the first part. Now let's go to the second part of our class, which is going to be male genital system. Okay, there will be lots of embryo. So I truly hope you remember your embryo. Actually, this self-control test, you will see a couple of embryo questions from last semester. So embryo 1, general embryo. Okay, so I truly hope you remember it. Because if you do, then it's easy. There's nothing to talk about. We already talked about it in embryo last semester. So male genital system composed of two things, composed of two testicles, okay? So it has two testes, and this is the area where spermatogenesis is going to be taking place. As you remember, spermatogenesis is production of spermatozoas. These spermatozoas are going to be traveling through a couple of ducts, and these ducts could be of two types. So we have the extra and intratesticular ducts. In addition, you will see three major accessory genital glands, such as prostate, seminal vesicles, and bulbourethral glands. They will be making something that will aid in the function of male genital system. And of course, the penis. Two primary functions of the testes are spermatogenesis, that's what we have just said, and steroidogenesis. Steroidogenesis means that you're making some kind of steroid, some kind of androgens. The androgen that you're making here specifically, this is of course testosterone. Now, maybe you know, maybe you don't, but originally testes are not present in the scrotum. As you know, scrotum is the sac where the testes are located, right? So it's just hanging outside. Originally, that's not the area where they are. Originally, testes are made in the abdominal body cavity, but then they descend down into the scrotum. They actually pass through the inguinal canals into the scrotum, right? Why is that? Why do they descend?
Gravity.
Okay, but what's the bigger reason? Why you don't want to have testes in the abdominal body cavity?
It needs to be in
Lower
the work
body temperature.
Yes. You all are saying the one thing, okay? The temperature. The temperature is the issue. As you know, your abdominal body cavity, or generally your body cavity, has a 36.6 degrees Celsius temperature. If you averaged out about 37, right? That's a bit too hot for your testes. So they need minimum 2 to 3 degrees lower, okay? So 35, 34 degrees, that's the optimal temperature for the testes to do their functions. My question to you is, if you have a boy, a man, his testes did not descend, so they're still somewhere in the abdominal body cavity, which functions will be impaired? Will it be both of them or just one of them, or like, what do you think? It will be spermatogenesis only. Only spermatogenesis function will be impaired if the testes do not descend. So for making testosterone, the temperature really does not matter. So for your self-control test, steroidogenesis could be happening at the normal body temperature. So you have a male, for example, whose testes did not descend. They're still in the abdominal body cavity, but this male still looks like a male. Why? Because he still has testosterone and is still being produced. Just one problem, this male will be infertile because spermatogenesis is impaired. You don't make spermatozoas whatsoever. So that's why you always check when the babies are born. You check, are the testes in the scrotum or no? How long will you give them to descend? Let's imagine baby is born and the testes are not there. How long would you wait to actually do surgery to pull them down? 1 year. Okay, so you wait 12 months. So you allow 12 months for the testes to actually descend, because in some boys this process is a bit of a late process. Okay, so you still wait 1 year. If after 1 year they didn't descend, you need to do a surgery. Okay, so you need to physically descend them down, like you do it as a doctor, as a surgeon. Okay, before we continue any further, we need to do a bit of anatomy. Okay, so here we have testes, right? They're in the scrotum. Scrotum is the sac where the testes are located. Inside of the testes, you have some kind of tubules. These tubules are called seminiferous tubules. So these seminiferous tubules is the place where spermatogenesis is happening, right? So let's imagine I made spermatozoas in my seminiferous tubules. So what is next? Where am I traveling from there? From the seminiferous tubules, spermatozoas are going to be traveling to the epididymis. And you will see that epididymis has 3 areas. So this is the head of the epididymis, this is the body, and this is the tail of the epididymis. So how are spermatozoas going? From the seminiferous tubules to the head, to the body, to the tail of epididymis. From the tail of epididymis, spermatozoas are going to the vas deferens. And do you see, it kind of turns backwards. So whenever you draw, remember, this is the epididymis like this, and then a sharp U-turn, this is your vas deferens. Okay, so here we have a testicle like this. So you have a sharp U-turn like that. Okay, so this is the epididymis, this is the vas deferens. Do you know where is the vas deferens located? How do we call a structure where it is kind of going? In anatomy, maybe you have learned it, maybe you didn't. Okay, it's called spermatic cord. Okay, so spermatic cord, that's like a tube-like structure which possesses lots of things, but one of the things that it has is actually a vas deferens. Okay, so here I have my vas deferens going up, up, up. It goes through the inguinal canal. It's entering into the abdominal body cavity, right? You can see that this vas deferens kind of loops behind the bladder. So it goes to the posterior surface of the bladder. And in this picture, it's not shown, but somewhere here, this vas deferens starts to dilate. Okay, so it has some dilated area right here. The dilated portion of the vas deferens is going to be known as the ampulla. Okay, so this area right here behind the bladder where the vas deferens is dilated, you will call it ampulla. As you can see, ampulla of the vas deferens is located right next to some kind of structure, which is called seminal vesicle. So the two areas, the two structures are very close to one area. They're in the same area, right? So ampulla of the vas deferens and seminal vesicle is right here. Both of the structures are behind your bladder. So here we have the seminal vesicle, and this vesicle will also have a duct. Also, there'll be some kind of duct that is coming out from this vesicle. The two ducts will fuse. So ampulla and a duct of the seminal vesicle, they will unite. And this fusion gives you another duct, which is passing through the prostate, which is called ejaculatory duct. So ejaculatory duct is, in a way, it's like a fusion of the two. It's a fusion of the ampulla, okay? It's a part of the fusion of the ampulla together with a duct of the seminal vesicle. So you can see this ejaculatory duct passes through the prostate and it opens up into the prostatic urethra, right? So you can see it here. This is prostatic urethra. So finally, your spermatozoas are here. They're in the prostatic urethra. What's going to happen next? Your prostate will make a fluid. You call this fluid seminal plasma. The seminal plasma will it will mix with the spermatozoas. So these spermatozoas are waiting for the seminal plasma to kind of be secreted here, and then they mix. This mixture is called semen, right? So it's a very common question in the world. They'll ask you, what is semen? Everybody knows that semen is the fluid that male ejaculates during sexual intercourse, right? So at the end of it, he ejaculates. So semen consists of two things. This is the seminal plasma fluid. Who makes this seminal plasma?
The prostate.
Prostate, yes. And it consists of spermatozoas. You have seen that spermatozoas really did travel long way, okay? So many ducts until they finally reached here. So how does ejaculation happen? Well, of course, here we have some kind of semen. Bladder is being closed by the two valves. So these two valves prevent the semen from going back into the urinary bladder. And through the urethra, which is located in the penis, right? Semen is going to be coming out into the vagina of the female, right? Now, what happens if male does not ejaculate? Okay, no sexual intercourse, but semen is still being made. Of course, I need to store this semen, right? So where is the semen going to go? The semen that you have just made is going to go back into the ejaculatory duct. And from the ejaculatory duct, it's going to go back into the seminal vesicle. So in a way, you could think that seminal vesicles are storage of the semen. Okay, that's the area where you will store extra semen, the semen that is not being used, for example. Let's imagine that seminal vesicles are also full. Okay, so I don't know, male really did not have for a long time. Okay, so where is the semen going to go? So seminal vesicles are full. Where is it going to go now?
Epididymis.
Exactly. It's going to return through the vas deferens into the epididymis. So you need to remember that specifically the tail of epididymis is the second most used area for storage. Okay, so you will see that seminal vesicles and tail of the epididymis. Okay, so testis. We have said that inside of it we have seminiferous tubules, right? Always is a question in the oral, draw me a seminiferous tubule. What can you find inside of it, right? So this is what, if you took embryo 1 with me, then you remember I have drawn this picture as well, okay? So several times. Seminiferous tubule has a lumen inside. So this is one seminiferous tubule. Inside of the seminiferous tubule, you will see triangularly shaped cells. These triangularly shaped cells are called, if you remember embryo, Sertoli cells. Very good. You call them supporting or Sertoli cells. So these are the triangularly shaped Sertoli cells. Why are they called supporting cells? Because they support spermatogenesis. They support production of spermatozoas. So actually, spermatogenesis happens between the two Sertoli cells. So if this is my Sertoli cell number 1, Sertoli cell number 2, the area between them, this gap, this is the area where spermatogenesis happened. Okay, who is responsible for it? So here we have some kind of stem cells. These stem cells are called spermatogonia. And there will be two types of spermatogonia, type A and type B spermatogonia. What is another name for them? Do you remember?
Dark and light.
Dark and light. So type A spermatogonia is a dark cell. Type B spermatogonia is a light cell. What is the difference between them? Type A spermatogonia will always, forever and ever, be a stem cell and nothing more. Okay, so the reason why males can produce sperm even when they're like 100 years old because of this type A spermatogonia, because it is always going to be a stem cell. What is this type A spermatogonia cell doing? It just gives rise to type B, and lets type B divide to become something else, right? Just give rise to another cell and let it do the division. Why should I do anything by myself? So type B will divide, and it will become primary spermatocyte. The division that we're talking about is mitosis. So through mitosis, type B divides to become primary spermatocyte. So in a way, you can agree with me that they will look the same because it's mitosis, right? It's exact replica basically. So you will see that they have a very similar appearance. Primary spermatocyte divides and becomes secondary spermatocyte. The division there is meiosis 1. Okay, so through meiosis 1, primary becomes secondary spermatocyte. Secondary spermatocyte enters meiosis 2, so it divides and becomes spermatid. Spermatids do not divide because there is no meiosis 3. Okay, so that's it. You finish meiosis 2, you're a spermatid. It simply matures and becomes spermatozoa. It just spends a couple of days in this stage until it eventually becomes spermatozoa. Actually, one of the major difference between them, right, is that spermatid is within the epithelium of the tubule. Spermatozoas are in the lumen of the tubule. But if you look at them, they look identical. They look the same. Just the only difference between them is the location. So as you can see, there are 5 stages, right? And these 5 stages are the stages of spermatogenesis. So what are the stages? Type A becomes type B spermatogonia. Type B gives you primary, then secondary spermatocyte, which gives you spermatid. Spermatid does not divide, it matures to become spermatozoa. So type, just generally, spermatogonia and primary spermatocytes. Are they haploid or diploid? Diploid, because mitosis. The way they divide is mitosis, so it's actually going to be diploid. Secondary spermatocyte is already haploid, right? So it's already going to be haploid. So starting from the secondary spermatocyte, all of these cells are haploid. So it is a question in the self-control test, which cells are diploid, which cells are haploid, right? So again, spermatogonia and primary spermatocyte are diploid. Secondary spermatocyte, spermatid and spermatozoa are haploid. Between them, we have a barrier, okay? There's a tight junction between them, between diploid and haploid cells. This tight junction is made by the Sertoli cells. This is why it's also known as the Sertoli barrier. Okay, so just a tight junction between the two Sertoli cells, Sertoli barrier. But you probably heard it as blood-testis barrier. Okay, so blood-testis barrier. This is your Sertoli barrier, and it's just a tight junction between two Sertoli cells. Yeah. So which cells are above this barrier? So think about it this way. Here will be basement membrane. So which cells are above?
Spermatogonia and primary.
They're at the base. Okay, so spermatogonia and primary spermatocytes are at the base, where the basement membrane is. So they're not above the tight junction, they're below it, because that's where a basement membrane is. Which cells are above this barrier? This is going to be the haploid cells. Haploid cells are above it because that's where the lumen is, right? So facing towards the lumen. Which cells are you trying to protect? Because it's a barrier, so which cells are you trying to protect? Diploid or haploid? Haploid cells. Why?
Immune system.
Immune system.
Amazing. And where is the immune system coming from? From here, from the basement membrane side. Somewhere there we'll have connective tissue. Connective tissue has blood vessels. Immune cells could come from this blood vessel, and they shouldn't see the haploid cells, because haploid cell is not a normal cell according to immune system. So it will try to destroy it, right? Okay, last step of spermatogenesis, so where spermatid matures to become spermatozoa, this is also known as the spermiogenesis. Okay, so don't forget that spermiogenesis is the last step of spermatogenesis. It's literally where spermatid matures to become spermatozoa. Between seminiferous tubules, because we will see that there are so many of them, they're everywhere, just within your testis, there is interstitium. So connective tissue, loose connective tissue, interstitium between seminiferous tubules. Within this interstitium, you can see some kind of blood vessels, and surrounding these blood vessels, we have interstitial cells. Another name for them is Leydig cells. So Leydig cells are going to be surrounding the interstitial, they will be in the interstitium surrounding the blood vessels. Leydig cells will be making testosterone. So a question for you guys. What is controlling this entire, all of it? Basically your testes, what are they controlled by?
The Leydig cells?
Leydig cells.
Okay, Leydig cells make testosterone. Who tells Leydig cells to make testosterone?
LH.
LH. And who tells LH to be secreted?
Hypothalamus.
Hypothalamus.
So GnRH. So which hormone controls everything here? Hypothalamus, but which hormone controls everything here? GnRH. Okay, so GnRH will be controlling everything here. As you remember, GnRH is made by the hypothalamus and it acts on the anterior lobe of the pituitary gland to secrete these two hormones, LH and FSH. So this is very important to remember that these gonadotropins, what does it mean, gonadotropin? It's a hormone which affects gonads. In our case here, the gonad is a testicle. So gonadotropins will be affecting your gonads. LH will be binding to the Leydig cell. Okay, so remember this, LH binds to Leydig cell. So the reason why Leydig cell actually makes testosterone is thanks to the LH. FSH will bind to the Sertoli cell. Okay, so FSH will bind to the Sertoli cell. And what will Sertoli cell do? Sertoli cell will secrete testicular fluid, and this fluid will be coating the lumen of the seminiferous tubule and making sure that none of the spermatozoas are actually stuck to it. Okay, it's just for lubrication purposes, this testicular fluid. But also very important to remember that Sertoli cell, thanks to the FSH, will also make a hormone. Also, they will make a hormone. And this hormone is called anti-Mullerian hormone. Without going too much into details of embryology, every embryo has two ducts. They are called Mullerian duct and Wolffian duct. Every embryo, doesn't matter, boy or girl, initially has two ducts, Mullerian and Wolffian duct. Wolffian duct will become epididymis, vas deferens, prostate, all of these male things. Mullerian duct will become uterus and fallopian tubes. So if I am a boy, which duct do I want to get rid of? Mullerian. I don't want in my boy to actually develop uterus and fallopian tubes. So that's why Sertoli cells have very important function. They actually make a hormone called anti-Mullerian hormone. And this anti-Mullerian hormone will be inhibiting the growth of the Mullerian duct to become uterus and fallopian tubes. In female, there is no such a thing as anti-Wolffian hormone. There is no such a thing. Estrogen does the job because estrogen is very active hormone. It's not like testosterone. You will see that testosterone is very lazy hormone. It takes so much time for it to actually do its job properly. So that's why there is a need for anti-Mullerian hormone. Anybody hablo español? Because fun fact, Mullerian is a female. in Portuguese and in Spanish. Okay, so Müller is a female in Portuguese and Spanish, so it's very interesting. Male is hombre, but which is also human. But yeah, okay, so remember that Müller, it's a female. Okay, so Müllerian hormone, it will lead to Müllerian duct, it will lead to formation of female things.
Julia, I have a question. So the Müllerian duct, do we get eradicated of, or do we just inhibit it?
Amazing question. So that's embryo right there. Okay, so you actually have a remnant of it. In embryo, we'll talk about it, but since we started talking about it, the remnant of the Müllerian duct in a male is going to be located somewhere here on the testicle, and it's going to be called appendix of testis. It's a very important anatomical structure. So in anatomy, you will learn it. So somewhere on the side of a testicle, you will find appendix of testis, and it is a remnant of the Müllerian duct. So thanks to this anti-Müllerian hormone, this huge Müllerian duct became so tiny. So it's just a little tiny structure called Müllerian duct, appendix of testis. In female, it's the same. So Wolffian duct in female does not disappear fully. It leaves two remnants. It's called apo-oophoron and para-oophoron. So soon in the anatomy, you will learn the structures. You need to show them as well. Okay, so appendix of testis, remnant of the Müllerian duct. Apo-oophoron, para-oophoron are remnants of the Wolffian duct. So they do not disappear fully. There are remnants of them. Yeah. Okay, so yes, lady, yeah, we talked. Okay, testes are surrounded by some kind of capsule. This capsule is called tunica albuginea. It's very, very, very dense, very thick. We will see it. There's also something called tunica vaginalis. It's not covering the entire testicle. You will only see it on the anterior surface of the testicle, just one layer of it on the anterior surface. Basically, tunica vaginalis is a remnant of peritoneum, because don't forget, testicles were developing in the body cavity, so just its anterior surface was covered by peritoneum. And as they were descending into the scrotum, they kept that peritoneum as tunica vaginalis. And you remember that peritoneum consists of mesothelial cells. So in a way, it's called mesothelial sac. Okay, this part, I don't think it's important whatsoever in the self-control test, but it will be important in the oral. So that's why we'll talk about it right now. Basically, why are you a female? That's the question that we should be asking. You're a female because on the short arm, okay, you're a female because you don't have the Y chromosome. On the short arm of the Y chromosome, there is a gene. This gene is called SRY gene. And SRY gene is standing for sex determining region of the Y chromosome. This SRY gene is coding for this factor, which is called testis determining factor. And as you can guess, if I have TDF, what will I have?
Testis.
Sex cords.
Yes, I will have sex cords, even better said, but testis. Okay, so testis is what I want right now. Okay, so you have testis. So why are you a female? Because you have ovaries instead of testis, and these ovaries are making estrogen. But why do you have ovaries? Because you don't have TDF. Why don't you have TDF? Because you don't have SRY. Where is SRY? On the Y chromosome. So you are a female not because you have XX chromosome, but rather because you don't have the Y chromosome. You're a mutant. Okay, so there is a theory that says females are mutants. We shouldn't be on this earth. Okay, but yeah. This gene, we'll talk about it in embryo, nothing important. This one is important, SOX9 gene. You remember AMH, right? Anti-Müllerian hormone. I forgot to mention it has another name, Müllerian inhibiting factor. It's another name, or Müllerian inhibiting substance. That's also another name. So AMH, MIF, the MIS are the same thing. They're all talking about anti-Müllerian hormone. So this SOX9 gene is responsible for its production. It activates the AMH gene. So the reason why Sertoli cell actually is making AMH, because it possesses the SOX9 gene that is actually being activated. This is very important part that you need to remember. As we have said, testosterone is very lazy hormone. Its half time is hours. I don't have hours, I need it now. So this is very typical question that they could ask you in the oral. Which androgen is the most active? Which one is the most fastest androgen there is? It's actually not testosterone, it's dihydrotestosterone. Dihydrotestosterone works like that, okay? So it binds to receptors, stimulates the processes like that. How can I make dihydrotestosterone? Testosterone has to be converted by this enzyme to dihydrotestosterone. This enzyme is called 5-alpha reductase. Okay, so this is Professor Rosa's favorite question, 5-alpha reductase. Every time she talks about it, every time, every year. Okay, so you need to remember that this dihydrotestosterone works on very specific organs, and that's PP, penis and prostate. Okay, so these are the two organs which have the most receptors for dihydrotestosterone. So in embryology, if they ask you this question, which hormone is responsible for the formation of penis? Don't say testosterone, because testosterone has nothing to do with it. It's actually dihydrotestosterone that has everything to do with penis formation. When do testicles descend into the scrotum, right? So when do they reach the final position? I love this because in the histo book, it says 26th week of gestation. In embryo book, it says 33rd, 33. So which one would you go with? Well, we're in histo, so 26. In embryo book, the number is different, 33rd week. Okay, temperature we talked about. Yes. Okay, this one we need to talk about pampiniform venous plexus. We actually kind of touched this plexus last semester. Basically, the structure is located in the spermatic cord. As you remember, inside of the spermatic cord, the most important thing is vas deferens. But right next to the vas deferens, there's also testicular artery. And this testicular artery is coming from the body to the testicles. So the temperature of this blood that is coming from the body to the testes is very high. It's 37 degrees, the normal body temperature, right? Testes are outside because they want a colder temperature. So do I want this hot arterial blood? No, I want it to be cooled down. So the purpose of this pampiniform venous plexus is that it kind of wraps around the testicular artery, and this allows some kind of heat exchange between the two vessels, because the blood goes in the opposite directions, right? So arterial from testicular artery goes from the body to the testicle. Pampiniform venous plexus, the blood is going from the testicle to the body, right? So how is the temperature of the venous blood? It's a bit cold because it's coming from the testicle to the body. How is the temperature in the testicular artery? Well, it's a bit hot. So it's perfect what we do. Pampiniform venous plexus wraps around testicular artery and heat is being exchanged. So by the time arterial blood reaches the testicle, it's already cooled down. By the time the venous blood in the pampiniform venous plexus reaches the body, it's already hot. Okay, so it's already 37 degrees. Okay. Have you guys been in Aquaticum before the corona? Have you been to this 37 degrees pool that they have there in the spa?
Yeah.
I always, when I go into this pool, I don't know, my stupid brain always associates with the temperature of my blood. And I'm like, oh my God, because it's hot, 37 degrees is hot. And I'm like, oh my God, this is the temperature of my blood in my body. This pool is the temperature of my blood in my body right now. It's freaking boiling. Okay, so that's why it's insane, like the association. So it's quite understandable why testicles don't want this blood, because it's freaking boiling. Okay, tunica albuginea we talked about. This is the capsule which is outside of the testis, right? So which is kind of covering the testis. It's very important to remember that this tunica albuginea, it sends septa. Okay, so it kind of sends septa into the testicle. And what we learned so far in histology, that if I send septa, I have a lobule, right? That's the main idea behind it. So is testicle lobulated? Yeah, in a way, there are little tiny lobules. If you count them, approximately 250, okay, of them will be there. But what's the most important thing about the septa is that you will find there a duct. This duct is called tubuli recti, or in English, straight tubules. And straight tubules receive spermatozoas from the seminiferous tubule. So if we ask ourselves, right, so what's the pathway? So in the seminiferous tubule, I make spermatozoas, but where do they go next? Next thing, they go to tubuli recti. That's where they go. And where is tubuli recti? It's in the connective tissue septa. It's inside of the septa. So that's a big help for us histologically, right? So we can actually identify septa. In it, we see a duct. This is tubuli recti. This part we talked about. Okay, so Sertoli cell. Sertoli cells have lots of names. They're also known as supporting cell or sestentacular cell. A couple of years ago, they named it. Doesn't matter. These cells do not replicate after puberty. So you have time until puberty to kind of make these Sertoli cells, and after that, you're just kind of left with them. They're triangular in shape. We have talked about it. In the book, it says that they cannot be seen with H&E. Like, do you see? It has no color, right? So it's kind of pale. That's what the book says. In the lectures, it says that they're eosinophilic. They say that they're eosinophilic because in our slide, they're actually eosinophilic, and that's really good help for us. So if you kind of look at the super eosinophilic cells, triangular in shape, you will know that these are Sertoli cells according to our department. Remind me, what do Sertoli cells do?
Testicular fluid.
Testosterone.
Leydig cells make testosterone. I heard testicular fluid. That's good. Okay, so testicular fluid is coating this lumen. Very nice. What else? Why are they called supporting cells?
Because from
The surface epithelium.
the process happens.
They come from surface epithelium, but it's not related to this one. It's a good point that they come from surface epithelium, but that's an embryo. So here it's supporting because it supports spermatogenesis, because spermatogenesis happens between the two Sertoli cells. And what is the last function?
So tight junction.
Mullerian.
I wanted Mullerian, but tight junction is also good. Barrier is also a function, but I wanted the anti-Mullerian hormone. So don't forget that it actually is in a way endocrine cell because it does make anti-Mullerian hormone. Which hormone tells you to do so?
Gonadotropin releasing.
Directly?
FSH?
FSH. Okay, so FSH. So, but you're right. Gonadotropin releasing hormone first, then FSH, and then only it will bind to Sertoli cells, so Sertoli cell can make AMH. Yeah.
Julia. I cannot see the triangular shape of
I
this.
know this is in the book. I will show you in our slide, and I will say it's a bit better. Like I keep saying triangular shape because in our slide it's much better.
Okay.
Okay, we talked about these spermatogenic cells. We talked so much about them. So two types, type A and type B, they're sitting at the base. Okay, so this area here is called tunica propria. So tunica propria is like basement membrane and lamina propria at the same time. So this area right here is not like two layers, it's just one thing. It's called tunica propria. And you could say it is in a way basement membrane and simultaneously lamina propria of this area. Inside of this tunica propria, we have smooth muscle cells. That's what's weird, because in lamina propria, we should have fibroblasts with collagen fibers. But here we have smooth muscle cells. And these smooth muscle cells are called myoid cells. You'll call them myoid cells. What's the purpose for these myoid cells? Well, they'll contract and make sure that spermatozoas within the testicular fluid are being propelled along the entire pathway, right? So they can move. Due to normal consequence of aging, tunica propria becomes thicker and thicker the older you get. Okay, so more and more tunica propria is being added the older you get. Okay, Leydig cells, we talked about them. Another name for them is interstitial cells because they are within the interstitium, right? So they're between the tubules. They are polygonal eosinophilic cells that make testosterone. Because they make testosterone, they have lots and lots of lipid droplets. Tell me why? How is it related?
Testosterone is a cholesterol derivative?
It's a steroid, and steroid has a cholesterol ring, right? So it has a cholesterol ring, so I need lipids. Because they have lots of lipid droplets, you will see that they also have lots and lots of smooth endoplasmic reticulum organelles. So as you remember from Histo 1, smooth endoplasmic reticulum is the area where we make and store lipids. Okay, so that's the area where we make and store lipids. Leydig cells, we have said, make testosterone, but it's very important to remember that this testosterone production does not begin at puberty. There are two times when Leydig cell makes testosterone, and the first time it makes so is actually in embryo. So approximately from 3rd until 5th month of fetal life, Leydig cells become super active and they make so much testosterone. And this testosterone is used for the development of the gonads in the male fetus. So you use this testosterone to make sure that testicles are nicely formed, that this testosterone could be converted to dihydrotestosterone, and then you will use this dihydrotestosterone to make penis, right? So it's understandable why would you make testosterone in the embryo, right? So you can definitely turn this embryo into a boy. Then from 5th month of fetal life until puberty, these Leydig cells are inactive. So they don't make testosterone, they are sleeping. And then when you hit the puberty, they actively start to make testosterone again. For which reason? For the secondary sex characteristics. What are these? So that's the enlargement of the penis, the hair, the voice, right? The muscle, all of these things are going to be there as well. So remember, first we make testosterone in embryo to make a boy, and then we make it at puberty to convert this boy into a man. Okay, so 2 times you make this testosterone. Okay, this part we talked about. Remember, Leydig cells are called interstitial cells because this is technically interstitium. Okay, whatever area is between the seminiferous tubules, this is actually called interstitium. So how would you call this spermatogonium?
Primary.
We have primary spermatocytes, but we don't have primary spermatogonia. So this is somebody was
Type
screaming.
B.
Type A.
Type A. Why type A?
Because it's dark.
Because
It's dark. What about this one?
Type B.
Type B.
Type B, because it's light. Do you see the difference between them? In our slides, it's even more evident. Okay, so our slides are really good. Okay, so it's even more evident. How would you call these cells?
Myoid.
Myoid cells, yes. Okay, we talked about this. Remember, which cell divides through mitosis to become something?
Primary.
Type B.
Spermatogonia divides.
Spermatogonia divide to
Spermatozoa.
become primary spermatocytes through mitosis. Primary spermatocyte becomes secondary spermatocyte through
Meiosis.
Meiosis.
meiosis 1. And secondary spermatocyte becomes spermatid through meiosis 2. Does spermatid divide?
No,
No.
no.
It matures. How do you call the process?
Homeogenesis.
Spermiogenesis. Very good. You'll call it spermiogenesis. Yeah. 2 cells, you remember, we talked about it. Okay, speaking of spermatozoa, according to histology, its length is approximately 60 micrometers long. According to embryology, it's 70 micrometers long. Which one you should go with? I don't know. Okay, obviously they won't make you choose. Okay, they will give you like weird numbers like either 70, 90, 200 or something like this, but they won't make you 60, 70, 80, 90, which is the length, right? So they won't do that. Just one of the two, either 70 or 60. So what does spermatozoa consist of? Of course, there is a head, neck, middle piece, principal piece, and the end piece. Together, principal piece and the end piece are known as the tail. Which kind of structure is the tail?
Flagellum.
Flagellum. What is flagellum? What is it?
Celia.
Amazing. It's actually cilium. It contains microtubules, 9 plus 2 arrangement, so that's why it's actually cilium. And you need to remember from Histo 1 that flagellum is actually the longest cilium there is. Okay, so it's super, super long. Yeah. In the middle piece, we have something here, which is called mitochondrial sheath, because that's the area where majority of the mitochondria will be located, right? So that's like a little tiny engine of the spermatozoa. So we make ATP, so this flagellum could be actually moving. Could you have mitochondria in the head? Well, of course, according to the book and lecture, you cannot. But fun fact, there are some Scandinavian families in Sweden, Norway, and Finland where males in those families, they have mitochondria in their head, in the head of spermatozoa. So which kind of DNA, which kind of mitochondrial DNA do the children in those families have? Maternal or paternal?
Maternal.
Paternal.
Paternal. But what's the normal way? What's the normal way should be? It should be maternal. Specifically because it's not normal. It's not normal to have mitochondria in the head. But in these families in Scandinavia, they realized that actually some families have paternal mitochondrial DNA. So that was like, oh my God, why is that? And then they realized that their spermatozoas actually do have mitochondria. And then as this research continued, they realized that more and more families actually have paternal mitochondrial DNA. So soon, I'm guessing soon, they will change this. They will say that it's not like, it depends. Some families do, some families don't, right? But of course, right now for you, no mitochondria should be present in the head of spermatozoa, that we inherit our mom's mitochondrial DNA. Here we have something called acrosome. Could you remind me from embryo 1, what is acrosome?
It's an enzyme which will help to
Very good.
dissolve
Yes.
the corona radiata.
Not corona radiata, but
Zona.
zona pellucida. Yes. So don't forget your oocyte is surrounded by a couple of things, right? So the first layer is going to be the zona pellucida, and then you have the corona radiata. So acrosome possesses digestive enzymes. So technically behind acrosome, you have digestive enzymes. And these digestive enzymes are needed to digest the zona pellucida, to make a hole through which the head of spermatozoa can kind of squeeze in, right? And so fertilization could happen. Don't forget that acrosome does not cover the entire length or volume of the head. It only covers anterior 2/3. So there is still part of the head which is not covered by the acrosome, right? How long can spermatozoa survive in the female's reproductive tract?
5 days.
Very good, 5 days. Okay, so it's exactly 5 days. How long is the entire spermatogenesis? 74 days. So from the spermatogonium to the spermatozoa, the entire process takes 74 days. But knowing that, every day a male makes approximately 200 million spermatozoas. So they're all going at different stages, okay? So every day. Okay, we have said that here we have seminiferous tubules, right? And now spermatozoas are going to be traveling through from these seminiferous tubules into the next duct, which is located in the connective tissue septa, which we call tubuli recti, right? So straight tubules. You remember that the only thing which is special about them is that they are located in the connective tissue septa. Simple cuboidal epithelium, nobody cares. From tubuli recti, okay, spermatozoas go into the rete testis. And rete testis are located in this particular area of the testicle. Okay, so this area right here. This area of the testicle is called mediastinum. Okay, so this is the mediastinum of the testicle. And this is where rete testis are located. Okay, so if they ask you where are they, they're in the mediastinum. Rete testes are known for their super irregular lumen. So if you see a tube and its lumen something like this, okay, so it's super, super weird. I don't even need to know anything else. Where is it located? I can tell you that's rete testis. That's the only tube in the testicle that has this irregular appearance. From the rete testis, we go to the next one, which is called vas efferent. Ductuli efferent. Do not confuse it with vas deferent. Okay, so it's not the same thing. This is going to be vas efferent. Vas efferent has two types of cells. Some of them are ciliated, some of them are not. So it's going to be quite helpful to you. Look at the epithelium. If you see some cells have cilia, and then right next to them, some of them do not have cilia, vas efferent. Now, starting from the vas efferent, this is the area where we start to absorb some of the testicular fluid. Because what you guys should understand is that these spermatozoas are not traveling by feet. They're traveling within testicular fluid. So if I have this seminiferous tubule, as you remember, Sertoli cell makes this fluid, right? We said it's for the coating of the tubule, right? But it's not just for the coating. When this tubule contracts to kind of push these spermatozoas forward, the fluid also moves. So until this point, until the vas efferent, spermatozoas are actually traveling within this fluid. So are they using their flagellum? No. Basically, it's like you're in the seas, catching wave after wave. You're just sitting like that and waiting until the fluid is just carrying you somewhere else. Okay, it's literally like you're on a boat in a river. River is moving, you're just sitting on the boat and just waiting until you get to the final destination. It's very important for spermatozoas to actually learn motility, to use their flagellum, okay, to make sure that they actually use the flagellum. And where do they learn the motility? Not in the vas efferent. They learn it in the head of the epididymis. It's just that you start to absorb the fluid in the vas efferent. So do you understand why do we need to absorb the fluid? Please don't make me repeat it.
Yes,
Why do we need to absorb the fluid?
To move a spermatozoa.
So they can
Learn how to
learn the motility, okay? So they can actually learn the motility function. Where do they learn motility function?
head of the epididymis.
Very good. At the head of the epididymis, okay? At the vas efferent, we're just starting to remove some of the fluid, okay? Just starting, so they slowly learn to move. Okay, these are intratesticular ducts. Remember this. Okay, so these are intratesticular ducts. So they're just kind of inside of the testicle. So from the vas efferent, we go to the head of the epididymis. head of the, basically not head, just entire epididymis, you remember it consists of 3 regions. So here we have the head, this is the body, and this is the tail. And if we stretch the tube that epididymis is, it will be 6 meters. And I'm going to shut up here for the dramatic effect. Okay, so if you stretch this tube, it is 6 meters. Dramatic effect achieved. Okay, so here it's super, super, super important. Okay, so you can see that it's super convoluted. This little tiny thing, which is actually about 7 centimeters length, like this, but it's so convoluted that if you actually stretch it out, it's 6 meters. That's insane. The pathways spermatozoas are passing through, right? What's the purpose of epididymis? Well, we have said in the head, we have to reabsorb this testicular fluid to make sure that spermatozoas actually learn how to move, how to use the flagellum. So what will I use for reabsorption? Take a look at its epithelium. In the epididymis, you see that there is pseudostratified columnar stereociliated epithelium. From Histo 1, you remember stereocilia are basically
Non-motile. I mean,
yeah, they're non-motile, but basically what are they?
mechanical sepals.
Microvilli. Basically, they're microvilli. So don't forget, what are we doing here? We're trying to reabsorb the fluid. So what do I need for reabsorption? I need microvilli. There's so much fluid, so I need super long microvilli. And these super long microvilli are your stereocilia. Okay, so you use the stereocilia for this purpose. Okay, don't forget in the tail, that's the area where we are going to be storing a bit of spermatozoas, okay? So extra ones could be stored here. From the tail, you can see we're making like a U kind of turn, right? And vas deferens begins. Vas deferens is actually also quite long. If you measure its entire length, it's about 60 centimeters, okay? 40 to 60 centimeters, that's a lot, okay? So it's a huge duct like this. How do you call its dilated portion here?
Ampulla.
Ampulla, very good. What is this?
Prostate.
Seminal
Prostate
vesicle.
is here, under the bladder.
Seminal vesicle.
Seminal vesicle, very good. You can see the 2 ducts are fusing together. And what is this? Ejaculatory duct. They look the same, actually. So if you do histological kind of view of both of these ducts, they look the same. It's just that they, like maybe the lumen will be a bit different because ductus deferens has a bigger lumen, but technically they're the same. Okay, vas efferent, we talked about it, so read it at home. For epididymis is the same thing. We talked about it, read it at home. You remember, right? Head, body, tail. So it's just a repetition of everything that I have said. Okay, this is important. This is the vas efferent. No, this is the epididymis, sorry. Okay, so this is the epididymis. You can see that these are the stereocilia that are at the top. But honestly, these stereocilia are nothing compared to the stereocilia that our slide has. Our slide has stereocilia like this, okay? So super, super long, super protruding like this. Once you see it, you know that you're looking at stereocilia, okay? 2 types of cells are in the epithelium. These are principal cells and these are basal cells. Of course, from the names, you can guess that the basal cells are the stem cells, right? Principal cells are the ones that actually have stereocilia. Everything else, just read it at home, we talked about it. Vas deferens, the same. We talked about what is it passing through, it's just extra information. So read it at home. So that's again, ampulla and seminal vesicle, right? This is your prostate, ejaculatory duct is right here. Now let's talk about accessory glands. So we have 3 of them. We have seminal vesicle, prostate, and bulbourethral gland. Seminal vesicles, in addition to storing the semen, they also make fluid. So as semen is being stored here, it also being added something to it. So seminal vesicle also adds something to the storing semen, and this something is fructose. Okay, so it's kind of making a bit more of this fluid, and this fluid possesses lots and lots of fructose. Of course, you remember, you know this, that fructose is the #1 source of energy for spermatozoas. What is the second source? #1, fructose. #2?
Mitochondria.
Hmm?
The mitochondria in the spermatozoa.
Mitochondria, they're just making the ATP. So it's just you're just kind of saying that they're making ATP. That's the process of making energy. But the question is source of energy. #1 is fructose. #2,
Glucose?
I could see why you would think so, but actually no. Surprisingly, ascorbic acid, vitamin C. Second source of energy is vitamin C. Your spermatozoas take up this vitamin C, break it down, and convert it to ATP thanks to the mitochondria. Okay, so that's the interesting thing. Okay, so seminal vesicles, as you can see, highly convoluted tubules. Okay, so there are lots and lots of kind of convoluted appearance here. That's the area where we'll be storing the semen. Here we have a prostate. Prostate will be making seminal plasma, as you remember, right? The fluid, which is the major part of the semen. This fluid will also be quite rich in fructose, ascorbic acid, vitamin C, some simple sugars, okay? So amino acids, all of those things, and most importantly, prostaglandins. Actually, fun fact, it's called prostaglandin because the first prostaglandin was isolated from the prostate. So they called it prostaglandin. But then they realized that not only prostate makes prostaglandins. As you know, there's so many of them. There's so many types of them. A couple of them are for the immune system, okay? But they're still called prostaglandins because the first one was isolated here. One of the prostaglandins that is being made here is needed for the smooth muscle contraction. So could you tell me how is this beneficial to have prostaglandins?
In the vagina.
Yes. So female reproductive tract is basically one huge smooth muscle. Vagina is a smooth muscle tube. Uterus is a smooth muscle tube, and fallopian tube is also a smooth muscle tube. So if you have this prostaglandin in the semen, this will cause contraction of said smooth muscle cells. And what will that do? It will kind of bring semen closer to the fallopian tube. It will actually kind of propel it closer to the fallopian tube. So spermatozoas don't actually have to pass through that long distance. The uterus and vagina and even fallopian tube will help it, right? If there is prostaglandin. Actually, fun fact, there are some females that actually experience this contraction very intensely right after sexual intercourse, where there is presence of semen in the vaginal orifice. These females claim that they actually feel for the next couple of hours how they have this cramps, okay, that their uterus or their vagina is actually contracting and bringing the semen closer to the fallopian tube. Do not forget that prostate will be making this fluid in a very particular pH, 7.29, okay, to be more specific, but generally just alkaline pH, because female reproductive tract is acidic. So we need to buffer it up a bit, make sure that this acid does not destroy the spermatozoas. Basically, prostate is like a tubular alveolar gland. Remember from Histo 1 or from first ACT, basically it has acini, and these acini are either tubular in shape or alveolar, right? So it has some ducts as well. So the same thing as any gland there is. If you will take a look at it, you'll actually see 30 to 50 of said tubular alveolar glands. But very important is that the stroma, right, so the connective tissue of the prostate gland does not only possess fibroblasts, which are making the collagen fibers. It also possesses smooth muscle cells as well. And we actually have a slide to show just how many smooth muscle cells are there. And you will see it actually has more. So if we compare number of fibroblasts and number of smooth muscle cells, we will see that stroma of the prostate has more smooth muscle cells than it does of the fibroblasts. Okay, so that's what I was trying to tell you here. Prostate could be divided into several zones. So we have, for example, central zone. Central zone is the area that is surrounding ejaculatory ducts. Central zone is the least likely to become cancerous. Okay, so it's the least likely one to become cancerous. In the periphery of the prostate, we have peripheral zone. Peripheral zone is the most likely to become cancerous. So as a doctor, you should remember that cancer of the prostate is never in the center. It's always in the periphery. Cancer of the prostate does not have any symptoms because it doesn't occlude any of the ducts. It doesn't occlude urethra. No symptom. So that's why screening is very important. You need to make sure that males older than 40 years old come for the prostate checkup at first every 3 years and then yearly, okay, to make sure that you can detect if peripheral zone is cancerous or not. Transitional zone is just where the urethra is passing, right? And here we will see normal physiological aging related issue with age. In the transitional zone specifically, there are more and more smooth muscle cells appearing here. So as you can imagine, if there's more smooth muscle cell, it will eventually obstruct the urethra. So with age, males have difficulties urinating. They have very much difficult initiation and very difficult to stop urinating because something constantly is pushing the urethra, and that's the smooth muscle cells which are proliferating here. Yeah, that's important. You remember we said that when we talked about the pineal gland, we said that there's a brain sand. Do you remember? How did we call the brain sand? Corpora arenacea. Okay, we call it corpora arenacea. And I told you that we have another sand if you're a male, and that's the prostatic sand, right? So this is the corpora amylacea. So remember this. So there's corpora amylacea. So it's also like calcium precipitation in the prostate. You remember DHT, right? Right. So dihydrotestosterone is 30 times more potent than testosterone. So it's way more active than testosterone. It will do its job amazingly. DHT is working on which things?
Penis and prostate.
Very good. So PP, so that's penis and prostate, okay? This part is not important. It's just some secretions that it also makes, but it's not important. Okay, bulbourethral glands. We talked about them. No, we haven't talked about them. Sorry. Too many groups are mixing into one. So we have here the prostate, we have here the urethra, okay, and this is the base of the penis. That's where the penis kind of begins. So you have here some kind of gland, which is called bulbourethral gland. Another name for it is Cowper's gland. Bulbourethral gland is making a mucus fluid, which will be clearing the urethra prior to ejaculation. Okay, so before a male ejaculates, you will see that there will be some kind of mucousy passage, like passage of a mucousy substance, and it is made by the bulbourethral gland. I truly apologize, but in everyday language, this is called precum. Okay, and you will see that in the doctor's office, you will see what I'm going to be talking about right now. Can you get pregnant from said precum?
Yes.
Absolutely. In Russia, we actually call it Virgin Mary's birth. Why? Again, truly do not mean to offend any religious person. It's just that Russia is an atheist country. Okay, so yeah, so that's why we're very disrespectful towards all religions, everybody. So we call it Virgin Mary's birth. Why? Because technically there was no intercourse, technically, but this mucousy substance, which was clear in the urethra, was present near the vaginal orifice, and it did get pregnant. It did impregnate a female. Why? You have to understand one thing. Spermatozoas will always be present in the male's urethra. Actually, fun fact, when you do lab work next year, third year, clinical biochemistry, you will, if you're lucky enough, you will see urine of a male, right? And you will look at it under the microscope at any time, any day, any, I don't know, point of the month, any winter, autumn, summer, doesn't matter. If you ask a male to give you a urine sample, in that urine sample, you will always find minimum 100 spermatozoas. Because this prostatic urethra and this entire urethra is a one passage for two things, for urine and for ejaculation, spermatozoas are present always there, okay? Always you will find them. So sometimes you will see that some girls use a very interesting method of contraception, which is, again, I'm really sorry, is called pull-out technique, where technically a male does not ejaculate into the vaginal orifice. Only this liquid substance is present there. And they get pregnant from that said substance because this substance is meant for clearing the urethra. But do you think it does it properly? Like with, I don't know, lots of tissues around, making sure that no spermatozoas are present there? No, it's just lubricating this area. But at all times, you will find spermatozoas in this area. So point of my story, this fluid, in a way, could impregnate a female. But of course, this topic is very sensitive because it depends on so many things, but it could, okay, so it could. Why is it mucousy? Because it has so many sugars. You'll take a look at it, right? So galactose, galactosamine, methylpentose, so many sugars are present there. So it's very viscous. So in a way, you could even say that it's not even for the purpose of clearing. It's just for the lubricating, to making sure that semen, when it's being passaged here, so it just does it very nicely. Okay, this is a pathway for you. Just do it at home. So the entire pathway, who goes where and how. So let's begin our slide here. Very important to remember that basically my slide here is upside down. So I will tell you what do I mean. It was my fault, but yeah, it's upside down. You will see. So the blue line here, this is the testis. So this is the testicle itself, right? So you can see inside of the structure, where many seminiferous tubules are present, okay? So many seminiferous tubules are going to be present right here. Between the seminiferous tubules, you can see that there's some kind of connective tissue septa. And this is a perfect example because within this connective tissue septa, you can see that there is a duct. So how would you call this duct? Tubuli recti, okay, so you will call it tubuli recti. So this is a straight tubule. This straight tubule receives spermatozoas from the seminiferous tubules, right? So from seminiferous tubules, we go to the tubuli recti. How would you call this area of the testis? It's like a middle area, so we called it mediastinum. Yes. We called it mediastinum. Okay, so we called it mediastinum. Within the mediastinum, you can see some tubules here, right? What are these? I told you that they're always present there. So these are rete testis. So from the tubuli recti, which are within the connective tissue septa, spermatozoas are going to the rete testis, to this area right here. Where do they go from the rete testis? Before epididymis?
Vas efferent.
Very good. Vas efferent, right? So we're going to the vas efferent. And vas efferent is here at the bottom of my slide. Is this normal position?
No.
No. So this should be up. So my slide is upside down. I realized it too late. So if you don't understand what I mean, so if you look at the slide, this is the top and this is the bottom. So you can see that vas efferent is here, it's at the top. But on my slide here, it's at the bottom. So this slide should be upside down, should be like this. Is it clear? From the vas efferent, we go to the epididymis, we go to the head of the epididymis. So this is the head of the epididymis right here. From the head, we go to the body, but you cannot see the body here. So you go to the next thing, which is the tail. So this is the tail of the epididymis. And then from the tail of the epididymis, you go to the
vas deferens.
Vas deferens. Remember, you make like a U-turn, right? And then vas deferens starts to ascend upwards towards the abdominal body cavity. Clear? Okay, you magnify a bit and you see that there is some kind of capsule here present. Which capsule is this? That's tunica albuginea. Okay, so this is the tunica albuginea here. What are these? Come on, guys, what are these? Seminiferous tubules, very good. This is connective tissue septa, inside of which you can find
Tubular recti.
tubular recti. Okay, how do you call this area?
Mediastinum.
Mediastinum. What are these with the irregular fluid? What are these?
Rectal testis.
Rectal testis. Yes. What is this?
Tail of the epididymis.
Very good. Tail of the epididymis, because it's upside down a bit, right? Okay, so this is the rest of the mediastinum with the rectal testis, right? Seminiferous tubules, tubular recti within the septa, and this is the head of the epididymis right here. So just trying to show you that it's in a way upside down. Okay, so we magnify. These are the seminiferous tubules, right? How do you call the area between them? What is this?
Interstitium.
Very good. Interstitium, as you can see, is loose connective tissue, which possesses some cells and some blood vessels. Which cells are these?
Leydig cells.
Leydig cells, very good, yes. Connective tissue septa, what is this?
Tubular recti.
Very good. Tubular recti, or in English, this is the straight tubules, right? So tubular recti or straight tubules. So from the seminiferous tubules, you go here. Okay, so how do you call this?
Tunica propria.
Amazing. Tunica propria. Inside of the tunica propria, you can see smooth muscle cells that are called myoids. Which cells are these?
Leydig cells.
Leydig cells, yes. Okay, so these cells right here, they are going to be Leydig cells. Okay, at the tunica propria, at the base, you can see triangular cells. More. Okay.
Yeah.
Okay, you can see triangular cells. Yeah, okay, so finally. Yeah, so as you can see, our slide is a bit better. Do you see? Here, perfect triangular shape. It's like, just try to see it, right? So it has a bit of a triangular appearance, like these ones as well here. So a bit triangular. I'm really stretching it, but it has a bit of a, like this one is perfect. This is a perfect triangular cell right here. So you can clearly see it there, right? Okay, these cells are Sertoli cells, yes. Yeah, so these are the supporting or Sertoli cells. Okay, at the base you see here some kind of cells. What are they? Spermatogonia. I point to this one. Which one is it? Type A, which is a dark spermatogonia, which is a stem cell. What about this one?
Type B.
Type B, which is a light spermatogonia, which is not a stem cell. Above them, you can see these cells. What are they? Primary spermatocytes. As you can see, mitotic division, so they look a bit the same. Okay, so these are primary spermatocytes. What about this one?
Secondary.
Is it? They look the same. Do you see? This is primary spermatocyte. So this one,
Primary
still primary. They can be on the top of each other, but they look the same. They are still primary spermatocytes. Then you move up a bit and you see cells like this, a bit smaller.
Secondary.
Secondary spermatocytes, yes. So these ones are secondary spermatocytes. Somewhere within the epithelium, you can see some kind of cells here that are very thin. Spermatid, okay? So if it is inside of the epithelium, this is a spermatid. And if it is in the lumen, it is called spermatozoa. So whatever cells we have in the lumen, these are spermatozoas. Okay, we talked about all of those things. So just refresh your memory, right? So spermiogenesis, you remember, blood you remember, function, you remember. So now your turn. How do you call this area? Tunica propria. Interstitium will be this one. Okay, so it's where the Leydig cells could be. Is there a Leydig cell here? Not really. Okay, so these are the Leydig cells. Blood vessels like arterioles are present, capillary right next to it, right? So this is definitely going to be the interstitium with the Leydig cells. This is a tunica propria. The cells here are myoid cells. Which cell is this one?
Leydig. No.
Leydig is this one.
Sertoli.
Sertoli cell. Okay, so this is the Sertoli cell, guys. Again, try to see it. Try to really see. Has a bit of a triangular appearance, like this one as well here. Okay, so this is a Sertoli cell. This one is also a Sertoli cell. Oh, look at this one I found. Nicely done. Triangular. Okay, triangular. So these are the Sertoli cells. Okay, what about this one?
Type A spermatogen.
But it has a bit of a pale center, so agree with me, it's a bit transitioning, right? So it's kind of already in the process, right? What about this cell? Sertoli cell. Okay, so this one?
Type
It's really light.
Type B.
Type B. Okay, so you see the lumen, the nucleus is really, really light. Yeah, okay. Okay, what about cells at the top of spermatogonia? What about these ones?
Primary.
Primary. Yeah, this is a primary. What about this one?
Also primary.
Also primary. What about these ones?
Secondary.
Secondary, very good. Okay. What about these ones?
Spermatid.
Spermatid. And what about in the lumen?
Spermatozoa.
Spermatozoa, yes, very good. Okay, so these ones are going to be spermatozoas. Okay, irregular lumen. So these are
Vasa. No.
Retetestis, yes. Okay, so these are retetestis. Cuboidal epithelium, nothing interesting. You remember that they're within the mediastinum, right? So most important thing, irregular lumen, retetestis. Now we're at this area, and you can see that there are two components, right? So this is a magnified picture of this area, and this is a magnified picture of this area. So if you look into this area, you see that there are some cells that possess some kind of apical domain modifications. And right under this apical domain modification, you can see there's a sharp red line. So this sharp red line should be basal bodies. So basal bodies are under the cilia. So the cells here, some of them are ciliated, some of them are not ciliated. You do not see it, right? For example, here, no cilia, but then you see that there are cilia. Where is this situation present? In the vas efferent. That means that this area where I magnified into, this is a vas efferent. Then you look into this area and you see these things. Remember I told you super long protrusions. What are these?
Stereocilia.
Very good. So these are stereocilia. Epididymis is the area where you start to have stereocilia. And actually, in a way, you should have stereocilia in the vas deferens, not efferent, vas deferent. You should have stereocilia, but they're not as big. Picture like this is epididymis. You won't, it's nothing else. It's only in epididymis you will see something like this. So if this is vas efferent, then what is this right next to it?
The head.
The head of the epididymis, because you remember that that's the way it goes, right? So from the vas efferent to the head of the epididymis. So we magnify these two slides, and what do we see? Notice some cells do not have cilia. Some do. Some do not, some do. So this kind of pattern that I do have, I don't have, this is very specific for vas efferent. That's the area where we start to absorb some of the testicular fluid. And then here you see that it's very evident that these are my stereocilia. Do you see how long? It's insane. They're super, super long. Okay, and actually, fun fact, you have this exact picture in your big histo room on the right side as one of the paintings. I think it's the first or second towards the board. Okay, so exact picture like this. So yeah, it's very special place. So this picture like this, you don't even need to see the bigger picture. You know exactly that this is epididymis. Okay, so because super long stereocilia, super long. How do you call these cells?
Principal cells.
Very good. And these ones are
Basal.
And that's it. So that's it for today. Okay, let me stop the recording.